Good day, this is the Professor, CBMD PhD. Today's Friday, May 5th, 2023. It is 5:05 and 18 seconds, a.m. Eastern Daylight Time. And today's research, gut microbes. So let's hop to it here. As to boost cancer immunotherapies, fighting power, look to the gut. Study reveals how gut microbes alter the body's response to cancer immunotherapy. Date May 3rd, 2023. Source: Harvard Medical School. Immunotherapy has redefined cancer treatment, but not all patients experience the same benefit when treated. New research in mice identifies how gut bacteria alter the body's response to a common form of cancer immunotherapy. The animal's gut microbiota lowered the expression of two immune molecules, one previously unknown, to play a role in cancer, using antibodies to play the activity of either molecule or the interplay between them, enhanced response to cancer immunotherapy. The findings can inform the design of treatments that boost the efficacy of cancer immunotherapy among patients with suboptimal response. One potent factor in treatment outcome appears to be an individual's gut microbiota, the trillions of microorganisms that live in the human intestine. The study done in mice and published May 3rd in Nature pinpoints how gut microbes enhance the body's response to a common type of immunotherapy known as PD-1 checkpoint blockade, currently used for the treatment of 25 forms of cancer. The research found that specific gut bacteria can affect the activity of two immune molecules, PD-L2 and RGMB, as well as the interplay between them. The work also showed that blocking the activity of either molecule or the interplay between them enhanced responses to cancer immunotherapy and optimize the body's ability to detect and destroy cancer cells. The engagement between PD-L2 and RGMB acts as a brake on cancer-fighting T cells, and our work shows that treatment with antibodies that block the interaction of PD-L2 with RGMB releases this brake, allows T cells to eradicate tumors. The study also identifies the molecule RGMB as a previously unknown accomplice in sabotaging the body's ability to spot and destroy tumors. RGMB, primarily known for its role in nervous system development, is also found on the surface of cancer-fighting T cells. Until now, however, no one knew it played a role in regulating T cell responses to cancer immunotherapy. If replicated in humans, the findings can inform the design of therapies that improve immunotherapy treatment outcomes, the researchers noted. The findings offer a critical clue into a complex puzzle, and in doing so, suggest concrete ways to enhance the potency of cancer immunotherapy and improve patient outcomes. We propose a new approach to overcome the resistance to the current cancer immunotherapies by learning from gut bacteria that help our immune system to fight cancer. As critical to cancer survival and spread is its ability to evade the body's immune defenses. Starting in the 1990s, researchers performed some of the critical early work that elucidated how cancer manages to do so. The research work focused on two molecules, PD-L1 and PD-L2, that reside on the surface of immune cells. The research work showed that when PD-L1 and PD-L2 interact with another molecule, PD-1, on the surface of T cells, the activity of T cells is kept in check. Under normal conditions, this interaction functions as a brake on T cells to ensure they do not mistakenly attack the body's own cells and tissues. The researchers discovered that cancer exploits precisely this safety mechanism to evade detection and destruction by T cells. Cancer cells do so by expressing PD-L1 and PD-L2 on their surfaces, engaging with PD-1 and training in T cells. Cancer immunotherapies that block the interaction of PD-1 with PD-L1 and PD-L2 release the T cells attack against cancer and are known as immune checkpoint blockade. As such treatments, currently used for 25 forms of cancer, have revolutionized cancer care but a subset of patients do not benefit from them. Since the advent of these treatments, researchers have been trying to understand why. The interplay between the immune system and the gut microbiota has been the focus of research work for many years. The laboratory has identified not only mechanisms of regulation, but also specific microbial molecules and microbial enzymes responsible for modulating the immune system. The notion that gut microbes can affect cancer immunotherapy is not entirely new. Recent studies have found tantalizing clues about the role that gut microbes play in immunotherapy treatment outcomes. Until now, however, a critical question remained unanswered. How? In the new study, the researchers used mice whose colons were seeded with gut microbiota from patients with cancer. Some of those patients had responded well to immunotherapy, while others had not experienced much benefit. As these animals' response to immunotherapy mimicked the treatment response in the humans whose gut microbes now lived in their intestines. As comparing the immune system profiles of the two groups of mice, the researchers identified telltale differences in various immune cells involved in cancer detection and destruction. The findings suggested that gut microbiota alter the immune cell's behavior and therefore responds to immunotherapy. As mice treated with gut microbes from patients that had themselves responded well to cancer immunotherapy had lower levels of PD-L2 on a class of immune cells known as antigen-presenting cells. As these cells play a critical role in rallying the body's immune defenses, they do so by patrolling the body for pathogens or tumors and presenting these foreign or abnormal proteins to T cells for destruction. And conversely, mice treated with gut microbes from patients with a poor response to immunotherapy had increased levels of the PD-L2 molecule. And to tease out the effect of specific gut microbes, the researchers treated groups of mice with broad-spectrum antibiotics which kill gut bacteria. Antibiotic-treated mice did not respond to immunotherapy that blocked the PD-1 molecule. These mice, however, had high levels of PD-L2, the other molecule break, that typically acts through PD-1. Animals that had a robust response to the same treatment had lower levels of PD-L2. Intrigued that PD-1 blockade did not work, the researchers hypothesized that PD-L2 acts as a brake on T cells, not through PD-1 alone, but through another molecular accomplice. The researchers turned their attention to RGMP, which the laboratory had previously shown that RGMP and PD-L2 regulated immune tolerance in lungs. When the scientists treated the mice that had not responded to anti-PD-1 therapy, along with antibodies that blocked RGMP, these animals experienced both an increase in cancer-fighting T cells and rapid overall improvement. The interplay between the microbiota and immune cells in the anti-cancer response just got clear, and with the identification of RGMP as PD-L2's molecular accomplice, we have another target for cancer immunotherapy. As further analysis showed that the interaction between RGMP and PD-L2 depended on both the composition of gut microbes, the researchers found that certain gut microbes can affect the levels of both molecules. Mice with cancer whose intestines have been seeded with certain gut microbes had levels of RGMP on their T cells 6 times lower than animals with microbe-free guts and responded to anti-PD-L1 or anti-PD-1 therapy. In comparison, mice with depleted gut microbiota did not respond to these treatments and had higher levels of RGMP on their T cells, especially on T cells that had infiltrated their tumors. Likewise, mice whose guts were seeded with microbiota from patients with poor treatment responses also had higher levels of RGMP, a finding suggesting that patients who do not mount a good response to cancer immunotherapy harbor higher levels of RGMP on their T cells, which in turn interferes with their immune cells' anti-tumor response. Disabling the activity of either PD-L2 or RGMP was sufficient to preserve T cells anti-tumor activity, and ensured a robust response to PD-L1 and PD-1 therapy. Remarkably, blocking the activity of PD-L2 led to a potent anti-tumor response in animals treated with another form of cancer immunotherapy known as dendritic cell therapy. The observation suggests that modulating PD-L2 activity holds promise for boosting the response to multiple types of cancer immunotherapy, altering the composition of the gut microbiota in different groups of mice revealed that one organism, C. cateniformis, suppressed PD-L2 levels and rendered immunotherapy more effective than mice with cancer. Given that the human gut is home to thousands of bacterial species, this microbe is probably not the only organism capable of regulating anti-tumor immunity. The findings suggest that specific microbial molecules can be harnessed in the form of small molecule drugs to augment the immune system's ability to control cancer. As such treatments could supplement or be an alternative to traditional antibody-based cancer immunotherapy. A small molecule approach would have the added appeal of being cheaper to develop and store and easier to deliver into the body. Small molecule medicines are generally given as pills, while cancer immunotherapy is administered in the form intravenously infused antibodies. The researchers caution that while their work reveals a critical piece of the puzzle, it is likely only one of several ways in which the immune system and the microbiome interact in cancer. There are likely many other ways in which the microbiome can affect cancer immunity in general and cancer immunotherapy in particular. With this work, they found a whole new way of looking at how the gut microbiota affects not only the efficacy of cancer treatments, but cancer immunity in general. And this is a professor of microbiology, bioinformatics, pathophysiology, and theater of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.